Thank you, Peter. Can everybody hear me okay?
Yes, we can.
Okay, all right, that's good. All right, well, good morning, everyone. Good afternoon to those of you in the Eastern time zone. And we'll go ahead and get started. This will be kind of an informational webinar explaining a little bit about the disease, some management options for you, and a little bit about how you can separate out Bur Oak Blight from some of the other diseases of oaks that may confuse you. So with that, we'll go ahead and get started. Bur Oak Blight, affectionately known as Bob, is caused by a native fungus that was identified by Dr. Tom Harrington at Iowa State University. They were looking at a malady they were seeing of oak trees with leaf scorch symptoms, and toward the end of the 2000s they looked very closely and identified this particular fungus, Tubacia iowensis, as a different species of Tubacia from the leaf form of the disease that we have known for many, many years. Once this disease was identified, everybody kind of started looking for where it was, and the map down on the lower left is showing the distribution as of about a year ago. So this is primarily a disease of the upper central Midwest, most of Iowa, Minnesota, Wisconsin. I am located in the Chicago metro area, and we've identified trees here in the Chicago area that have been positively identified as Bob. This disease has two forms, and this is fairly recent to me. I attended the Shade Tree Short Course up in Minnesota in the middle of March and heard Dr. Harrington kind of give an update on this disease. So we'll talk about this in a little bit later, but there are leaf and petiole twig forms of this disease. Originally, we kind of thought there was just kind of one form that had both leaf and petiole that would attack both, but now it looks like there are kind of separate leaf and petiole twig forms of this particular disease. And the disease itself, as well as some of the associated secondary pests that may associate with this particular disease, can result in foliar necrosis, twig death, dieback, and even possible death of the tree. Bob is primarily a pest of bur oak, Quercus macrocarpa. Dr. Harrington did report that they have also seen some foliar lesions from Bob on swamp white oak, but those infections were on trees that were growing very close to other bur oaks that had the disease. So primarily we're looking at this as a disease of bur oak, but apparently it can also show up on swamp white oak, but he didn't really make a big deal about that. He thought it was mainly because they were in close association to infected bur oaks. There are two forms of Quercus macrocarpa, or bur oak. There is an upland form, which tends to be the savanna form that is very important to us in habitat restorations where native prairies interact with the natural forests in the upland or in the upland areas in the upper Midwest. So bur oaks, the upland form of bur oak, which tends to have the smaller diameter acorn compared to the other form of bur oak that we call the lowland form. The upland form seems to be more susceptible to Bob, and at least in the areas that I'm familiar with, the upland form tends to be the form of bur oak that is often found in the park systems as well as in some of the urban residential areas where bur oak is found. The lowland form of bur oak has the larger acorns with the large frilly cups whereas the upland form has a little smaller acorns and the cup isn't quite as fuzzy. It seems that Bur Oak has a wide variability in their resistance or susceptibility, and that's pretty characteristic of a native disease, and that is why Dr. Harrington believes this is a native disease that has become a little bit more prominent in the last few years. As I mentioned earlier, Bob seems to have two distinct forms. The first form is what we'll refer to as the foliar form. This form of the disease begins with small leaf spots on the leaf blade and eventually develops into the somewhat characteristic symptoms of the disease with these black lesions that will form on the midvein as well as some of the major side veins. So in these two photos you can see the foliar form with some of the leaf flecking and then these lesions that will begin to develop on the main veins. These lesions will then tend to show up fairly early in the season, so early to midsummer, and those lesions generally will grow together, and that will lead to some of the foliar symptoms that we will see a little bit later in the season. So the initial forms are a leaf spot that will move on to the main veins and grow together to essentially begin to block the translocation system in the leaf itself. So one of the reasons that we have seen more of this disease recently, it looks like, is going back to some of our spring precipitation. Now, this particular graph was developed, was pulled from the website provided by the SCIPP, which stands for the Southern Climate Impacts Planning Program. And I found it to be a very handy site to reference because you can pick your state as well as different regions and get historical temperature and precipitation data for the area. So in this case, since most of the research was done at Iowa State on this particular disease, I grabbed the historical weather data for central Iowa, where Iowa State is located. And you can see with this precipitation data, the gray lines indicate the actual rainfall compared to the historical average beginning in the late 1800s. The area under the curve represents precipitation data that's in 5-year trends. So essentially you're looking at the reference point at any year plus the 4 preceding years. So each year you move forward, the last year drops off, and you add the current year. So we could see that at least in central Iowa, and this is fairly representative of most of the areas of the upper Midwest that I've checked. You can see that since about 2000 or even in the late 1990s, we've been having above average spring rainfall. And it's believed that rainy periods during the late spring and early summer determine the severity of the foliar form for that season. So this is going to be similar to many other foliar type diseases that we can work with, whether it be other foliar diseases of oak, such as Tophrina or oak anthracnose, as well as many of our other foliar diseases of other plants, such as apple scab rust, that we know that rainy spring weather can increase the amount of disease we see from season to season, depending on how that spring rainfall pattern occurs. So this foliar form of Bob then appears to be associated with rainy periods during the late spring and summer. We begin to get those leaf spots that show up and begin to form those lesions on the midveins and major secondary veins that lead to these larger lesions. And when that occurs, later on in the season, we tend to get these somewhat anthracnose-like symptoms, these blotchy brown areas that form on the foliage, because once those leaf or those vein lesions kind of coalesce, that will essentially shut down the water nutrient transport from that vein and will essentially cause the distal part from that point to desiccate and brown out. So we end up with these wedge-shaped discolored areas on the leaf, and of course they're going to vary depending on where in the leaf they may form. So we may see some forming just on the wedges of the secondary veins, or we can see them forming on the wedges of the primary vein. They could be toward the end, they could be toward the middle, or toward the base of the leaf blade, depending on where that vein became obstructed. So this symptom, these wedge-shaped necrotic areas, tend to develop a little later in the season, in July and August. If we were comparing that to oak anthracnose, we realize that is a spring disease, which tends to show up in the early spring. So that can be one means of separating oak anthracnose, which is going to be a spring disease, from the foliar stage of bob, which is going to be developing later on in the season compared to oak anthracnose. The diseases tend to be heavier on the lower and inner portions of the canopy. Once again, that would be similar to a foliar type disease, very similar to what we would see with oak anthracnose or other types of foliar diseases, where the lower parts of the canopy, because of the increase in shade and less air circulation, isn't going to dry out quite as rapidly as the more exposed portions of the upper canopy, where more sunlight, drying conditions will occur, so there wouldn't be as much expectation of foliar diseases on those locations. The other thing about the foliar form is that many of the leaves infected with the foliar form will tend to drop from the tree. So this is a little bit different too. We originally thought the foliar form would grow down the petiole and contribute to the basal petiole form of the disease and the twig form of the disease, but it appears that many of these leaves with the foliar form tend to upsize and drop off the plant rather than the fungus growing down into the basal part of the leaf. And the, oops, let me go back. The foliar form is kind of considered the less important of the two forms compared to the endophytic form, which we will talk about next. So the first form was the foliar form. The second form we'll refer to as the endophytic form, and this form will kill the petiole base of the leaf. It really doesn't have any specific foliar symptoms. The foliar symptoms tend to be associated with the foliar form of the disease. The leaves that are attached to infected petiole bases don't fall from the tree in the fall. They tend to hang on. So if you look at a bur oak that has a number of these endophytic forms that are killing the petiole bases, the leaves don't drop. They tend to hang on the trees in groups. And so during the winter time, you can look up at the tree and see these clusters of dead leaves that didn't upsize from the canopy of the tree. They'll hang on through the winter, and then looking in the spring of the year, early spring, we have these clusters, and the fruiting structures of the endophytic form on the petiole bases. The petiole bases will often swell, and then we will get the fruiting structures that form on these petiole bases that are still attached to the tree. So oftentimes this dead petiole base, when we get these fruiting structures, this provides the spores that in the spring will blow off and infect the foliage, leading to the foliar form. So these spores serve as the source of the inoculum for the foliar form in the early spring, and then this form will also grow down and it'll grow into the twigs, and then when, because these stems are dead, the tree will often form new buds at the base, and then those new buds will grow new shoots, and those shoots can then be infected by the foliar or the endophytic form that can simply move down and up into the nearby shoots as they develop in the spring. So the endophytic form grows down and infects the new petioles of new shoots that develop as a result of the previous year's shoots being killed, and that tends to perpetuate the endophytic form of the disease from year to year. And the endophytic form is considered to be the more serious of the two forms. Rainy periods during spring bud break determine the seasonal severity of the endophytic form. So it seems that as these buds begin to grow and we get the spring rainfall right as the new foliage is expanding, rainy periods during that time frame can tend to increase the amount of the endophytic form. So spring rains in mid-spring, late spring can contribute to the foliar form, but rainy periods during bud break and leaf expansion can contribute to the severity of the endophytic form. So this discussion of the leaf form and endophytic form was new to me in hearing Dr. Harrington speak about the disease during the Minnesota Shade Tree Short Course up in Minneapolis that I attended. Some other observations relating to bob: it appears to be less common on well-drained soils and worse on clay soils. We know that oftentimes urban soils can be challenged in terms of poor soil conditions, so that wouldn't necessarily be so surprising. And it also appears that secondary pests such as two-line chestnut borer, armillaria root rot, that are often responsible for the ultimate decline of oaks once they enter a decline spiral, are most responsible for the branch dieback decline and death, which is also a little bit different. As we learn more about bob, we seem to see that the bob tends to be a precursor and may weaken the trees and make them more susceptible to other secondary pests that come along as the trees being weakened, as a result of bob or any other foliar type disease. So bob is acting as an additional stress factor, leading to increased susceptibility of those trees to the secondary pests that are often responsible for the more major decline symptoms in the oak tree itself. So looking at bob management options, general recommendations go, if you suspect bob or have a positive identification of the foliar of the fungus responsible for bob, at least in the initial stages, treat for the secondary pests. So if you feel a tree is under stress, treat for two-line chestnut borer. This could be done with some of our soil-applied systemics such as dinotephurin or metocloprid. And you may also consider paclobutrazol in the form of cambistat and some other trade name products to help promote root system development. And these two items can be general recommendations for stressed oaks, whether they may be suffering from bob or any other particular malady. And then proper cultural practices, proper watering in times of drought, nutrient management, mulching, soil modifications, treating compacted soils with air tools, or anything that is going to promote the overall soil health of the particular tree. Propiconazole in the form of Alamo has been recommended as a direct treatment for bob, especially in some of the literature that was coming out around the late 2000s, 2008, 2009, 2010. After a little bit more experience, it kind of can appear that propiconazole can have somewhat mixed results in terms of success. Paclobutrazole can also be phytotoxic on bur oaks. The photos that you're seeing here are showing some foliar leaf burn and discoloration as a result of Alamo injections that have been done on bur oaks for oak wilt. Paclobutrazole is propiconazole is a recommended treatment for white oaks that have positive diagnosis of oak wilt. So we tend to be seeing a little bit of a backpedaling about initial treating with propiconazole and not to necessarily treat with trees with low levels of disease with propiconazole, but perhaps treat for the secondary problems such as two-line chestnut borer or with cambustat in order to promote some extra root development that could be leading to some of these secondary problems. You may want to consider injection with the situation if it continues to worsen. If you monitor a tree for a few years and it seems to be struggling with continued infections of bob and it appears to be impacting the tree, you may want to consider an injection at that point. If you would consider a trunk injection, the Alamo is injected typically by a macro infusion process. You would want to do a spring application. You would want to use a lower rate of propiconazole. Generally the 10 mil rate is recommended. Do realize you may see some phytotoxic reaction in the tree even at that 10 mil rate. And then they suggest do not re-inject unless the disease continues to worsen. So, what are some lookalikes that we might run into? Bob is a form of Tubacia, but it's different than the form of Tubacia that causes the leaf spot on oak in late summer. This red oak leaf has the foliar symptoms of Tubacia. Oftentimes it's the foliar Tubacia. Oftentimes you'll see these leaf spots, oftentimes with a white center in the center. This is a red oak. Red oak does not appear to be susceptible to the Tubacia that forms bob, but it is susceptible, as well as the other oaks, to the Tubacia leaf spot that we knew existed for many years. And that is just that the bobs seem to be behaving differently than the foliar forms of Tubacia, and that led Dr. Harrington to wonder if we were dealing with the same disease. We also have the Tephrina leaf spot that will form on oaks in the spring. This kind of causes this little blotchy, sunken lesions. They almost become kind of like a pock in the leaf blade, but it's a spring-forming disease that'll typically form on oaks of all types in the spring of the year when we have those conditions that are favorable to foliar diseases of all types. And then we mentioned oak anthracnose that is present on all species of oaks as a spring disease, and it makes these irregular blotches that tend to cause leaf deformation and some crinkling of the leaves and can lead to defoliation. The main difference is the Tophrina leaf spot and oak anthracnose diseases are spring diseases, whereas the foliar form of bob is a mid to late summer disease once we start getting the leaf lesions, large leaf lesions beginning to form. Bastian spot is also a late summer developing disease, but is more of a leaf spot disease. And even though here it looks like it may be kind of getting into the leaf veins, it is a different disease than the leaf spot, than the bob form of the leaf fungus. Probably a disease that could be most confused with Bur Oak Blight is oak wilt. Now, oak wilt is shown here on the left side of your screen. The bob symptoms, the foliar symptoms, are shown on the right side of the screen. So once again, oak wilt tends to form lesions and discoloration, not necessarily lesions, it's more of a discoloration of the entire leaf blade. The foliar symptoms can be basal, they can be distal, they can be peripheral. You get a lot of leaves forming, as you can see in the background of this photo. A lot of leaves drop from the tree. Oftentimes you'll find green leaves dropping, half green leaves dropping, brown leaves dropping. You get a variety of leaves dropping. The Bur Oak Blight discoloration, once again, in many cases you tend to find this wedge-shaped discoloration on many of the leaf blades because the disease is actually causing a discontinuity in the main veins of the plant. The other thing with oak wilt, because it is a vascular disease where the oak wilt is actually getting into the vascular system of the tree and growing down through the vascular system from the top part of the tree into the bottom of the tree, or through a root graft into the lower portions of the tree and then spreading up. The symptom development tends to be top down and from the outside in on infested trees, because we're dealing with a vascular disease, and so we tend to see those symptoms showing up on the outer portions and working down toward the inside of the plant. The foliar symptoms of bob, on the other hand, tend to be forming in the more humid parts of the tree, which tends to be the lower canopy and the inside leaves, and they tend to work outward. So looking at how the disease moves within the tree, or how the symptoms develop within the tree, can give another clue to the difference between oak wilt and Bur Oak Blight when you're just looking at the tree, assessing the visual symptoms. Of course, there are other differences between oak wilt and Bur Oak Blight as well. Oak wilt is a vascular wilt, so as we just mentioned, it gets into the systemic part of the tree through the vascular system and can spread through the entire tree. Bur Oak Blight is a leaf and twig blight disease. It is not systemic in that it doesn't grow through the whole tree's vascular system. It tends to be restricted to small twigs and can cause some of that clustered twig death. But the more severe symptoms associated with Bur Oak Blight tend to be, we suspect, from more systemic issues such as two-line chestnut borer or the effects of armillaria root rot that can have these somewhat discombobulating effects on the trees in terms of nutrient and water movement. Oak wilt is spread via root grafts from tree to tree. It can be spread overland by insect vectors such as bark beetles or sap beetles. When we're talking about Bur Oak Blight, we're really talking about the spread of airborne spores similar to most foliar type diseases, and then fungal growth into the new petiole. So the disease is spread a little bit differently. Oak wilt will attack all oaks, but is much more serious on the red oak group compared to the white oak group, which does include the bur oak. Bur oak blight tends to attack only bur oak, especially the upland variety with the small acorns. Did mention a little bit at the beginning of the program, there have been some foliar symptoms found on swamp white oak. However, those were thought to be somewhat unusual because the trees were closely associated with bur oaks that had the symptoms. Oak wilt symptoms appear top down and outside in during the mid to late summer as wilting, variable leaf coloration, heavy leaf drop. This is going to, oak wilt is going to show up once you begin to get heavy transpirational stress on the tree because the bur oak or the oak wilt fungus is essentially clogging or causing the tree to shut down its vascular system, and anything that promotes increased transpirational water loss is going to make the symptoms of oak wilt show up even more. With bur oak blight, our symptoms appear bottom up and inside out. Early to late summer is black spots on leaf veins and on the leaf blade, progressing to wedge-shaped leaf blotching with some leaf drop in late summer. We don't have any vascular streaking like we will have in oak wilt. If we peel back the bark, we oftentimes have vascular streaking, which is the tree's response to the presence of the fungus trying to wall it off. We don't have that with bur oak blight. Infected leaf clusters can persist in the winter, and then small fruiting structures on the petiole base, as we saw earlier in some of the slides. Oak wilt is fatal to red oaks in a few weeks or months, and it can be fatal to the white oak group after several years. It tends to be more of a chronic, debilitating disease on the white oak group, but is typically very quickly fatal to the red oak group. Where bur oak blight is considered more of a debilitating disease and tends to predispose the tree to secondary pests such as two-line chestnut borer, armillary root rot, and some of the other issues. Oak wilt trees are often grouped in pockets because the disease is spreading through the root systems to nearby trees through root grafts, or in bur oak blight, the trees can be random and scattered, a lot of differences in susceptibility between individual trees, so you can have heavily infested trees right next to trees that aren't showing much of a symptom at all. This can be very similar to what we see in oak anthracnose, with two trees side by side, one with very heavy anthracnose symptoms and the one right next to it showing very little anthracnose. So with that, that kind of goes through the information that I have, and we would try to happy to answer any questions that you may have.
All right. Well, thank you, Dr. Bastian. And as Dr. Bastian mentioned, we're going to move on to the Q&A portion of the webinar now. I'll be reading the questions from the audience. Dr. Bastian, Kent Honnell, and Chris Haugen are all available to answer your questions. Please continue to write questions if you have any during this portion. So I'll just go ahead and see what kind of questions we have right now. Looks like we've got one here. The question is, where on the Alamo label does it state that it is acceptable for bur oak blight to be treated? I only see oak wilt, Dutch elm disease, and a couple of diseases listed.
Yeah, the research that was done at Iowa State looking at Bob, when they were looking, they were treating with Alamo as the primary source because it was already labeled for some of the oak maladies. So that is why the recommendation has been going out. So you would probably need to check with your local extension or Department of Agriculture to see how they look at some things like this. There are some Section 2 EE labels of FIFRA that some states are a little bit more liberal with compared compared to other states. So that would be something that you would check. Perhaps some of the folks there could comment on some of the state, how some of the different states were looking at this.
Yeah, and Rex, this is Chris here. Another part is on the Alamo label, there is in the label there is a opening which says it may be used for other diseases and on plant material not explicitly stated on the label, so long as you have done your due diligence that it will not be phytotoxic and that it has shown efficacy against that disease. And certainly with that that information done at Iowa State and that on the Alamo label, it would fall under that section. To further clarify.
Yeah, do remember that the Alamo can be phytotoxic on
Right.
on bur oaks, so you always want to once again have good client expectations, and I think that's another reason why they're kind of changing a little bit compared to some of the recommendations that were first coming out.
Yep.
Okay, looks like we got another question here. How many milliliters do you recommend for applications?
I think I mentioned that usually I would probably recommend beginning with a 10 mil rate. I believe Dr. Harrington said in his presentations you drop below 8 mils and it didn't seem to have an effect in some of their work. Some of the higher dosages that they tried resulted in more severe phytotoxic reactions. And I think some of the publications that showed some of that research data is available at Dr. Harrington's website via Iowa State. But it kind of appears they're saying using some of the lower rates rather than the higher rates to avoid the toxic phytotoxicity reactions. Also remember too that when you do trunk inject bur oaks, a lot of the sites will flux, so that's one of the other somewhat downsides to doing trunk injections on oaks that you have to consider.
Okay. Another question here. The map at the beginning of the presentation showed that this is primarily found in the upper central Midwest. Has it been spotted outside of this area? Example would be in the East Coast.
That is the most recent map that I have seen, and that was dated 2015. Now, obviously, if you went and really started to look, you could find it other places, but that is the last map that I have seen.
Okay. Another question here. If the infection is severe, should I include a foliar spray the first year? And if so, what should I use and what is the timing?
Any foliar sprays on large trees can always be somewhat problematic to get done because of the drift concerns. So you'd have to take that on a case-by-case situation, looking at the ability to spray some of these large trees with high volume pressure equipment. If you were going to, because the disease is a tophrina, you were looking for materials, I would check to see if any material that would actually have tophrina on the label, because tophrina has been an established disease on oak for many years. So you should be able to find some of the fungicides. I don't have something that's right off the top of my head, but I would generally probably look at some of the more common active ingredients such as thiophanate methyl or the sprayable forms of propiconazole like Banner, Max, as a place to start.
Okay. Another question here. I have heard that treatment earlier in the summer is better with Alamo, but the label says that treating oaks from May 15th to June 15th will attract insects that transmit oak wilt. Can I treat before June 15th?
Dr. Harrington said that spring applications are preferred if your target would be bob. Now, generally, we would want to wait because our absorption of the material is typically done by the transpirational pull. Generally, we would want to wait until we had full leaf expansion on the canopy in order to get the most draw, so you would have the most effective uptake in your injection procedure, whichever you would choose. So I would definitely want to wait until I had full leaf expansion before doing any injections.
Okay. It looks like there's another question here. I was late in joining. Can I go back through the slides still? To answer your question, this webinar is being recorded, and so we'll definitely provide the link to view it, and so you'll be able to see the presentation through its entirety. Okay, doesn't look like there's any other questions, so we'll go ahead and start to wrap up here. If you do have any follow-up questions or would like more information, certainly you can reach out to your territory arborist, go to our website, treecarescience.com, or shoot us an email, info@treecarescience.com, or you can call the Solution Center, and anyone here would be able to assist you. Before we end, I just wanted to make a quick plug about a very important project that we are working on. Saluting Branches is an annual event that we started last year. This year we're a nonprofit organization. On September 21st, 2016, we will be bringing together tree care and landscape professionals from all across the country to provide a day of service at local veteran cemeteries. Last year we had over 1,000 volunteers and donated over $1 million worth of tree pruning, removals, plantings, and plant health care. If you are interested in signing up this year, please go to salutingbranches.org for more information. And finally, I just wanted to mention that we do have some more webinars coming up. We've got some really great topics and presenters. If you would like more information, you can go to our website, treecarescience.com, and sign up and register for these if you haven't already. Oh, looks like we actually do have one more question here coming in. Besides injections and sprays, do you suggest any other treatments for bob?
Not, bob directly. Remember, once again, my approach would be to begin treatments treating for the secondary pests. So in addition, I would begin treatments with treatments for two-line chestnut borer, probably also with growth regulators and cultural practices to really, try to break the tree out of a potential decline spiral. I would do those things before I would start to consider a trunk injection for bob itself. Obviously, because of the differences in how spring and summer rainfall can impact this disease, we'll probably see the disease being worse in some years than other years based on what type of rainfall patterns we do have from year-to-year basis. So I would always begin with cultural practices that we may find available to us: proper water management in times of drought, alleviating compacted soils, mulching, surface composting, nutrient management as necessary, then some of the secondary treatments for secondary pests, and then if we still saw the disease being problematic, that's when I would consider the direct treatments for the fungal disease itself.
It also looks like there's a follow-up question here. Where does the two-line attack?
Two-line chestnut borer is an Agrylus species, so it's very closely related to bronze birch borer, emerald ash borer, and honey locust borer, native honey locust borer. So the initial attacks begin in the upper canopy, which is oftentimes you begin to see that stag heading symptoms showing up on trees that are coming under attack from two-line chestnut borer. And then in successive years, the beetles begin to move down the trunk as the crown begins to die back. They begin to work into larger and larger wood down the trunk, eventually getting down to the main trunk itself. So the Agrylus species all tend to work that way, which is typically why we see the initial symptoms of the borers up in the upper crown progressing downward in successive years. So in order to get good management with soil applied materials, you have to have an intact vascular system to move those materials up to where the borers are feeding. And the more damage you have to the vascular system of the tree, the less effective those treatments will be, simply because the chemical can't move up to where the insects are feeding because the destruction of the vascular system inhibits that movement.
All right. Well, thank you again, Dr. Bastian, for your presentation, and to Chris and Ken for sitting in to provide answers during the Q&A portion. One last final note, it would be much appreciated if you could fill out the quick survey that follows immediately after this webinar. We're always looking here at Rainbow for ways to improve on what we do and how we host these educational events. Again, thank you to Dr. Bastian. Thanks again for all of you for joining us today, and have a great rest of your day.